Well, thank you very much, Christoph, for that wonderful introduction. And I also just want to give thanks to the Rachel Carson Center for the fellowship that has brought me here and for creating such a congenial environment for writing and for thinking. And it's just really great to be here. And I think all of the students who are associated with LMU and the Rachel Carson Center are very fortunate. Okay, so today I'm going to talk about my research project here at the Rachel Carson Center. It's a project, in a way, in global environmental history that's concerned with the oldest environmental pollutant that human beings have had to deal with, which is our own human waste. So the project deals in part with our efforts historically to deal with it, how to keep it from sickening us and killing our children, how to dispose of it, whether or not to consider it a renewable resource, whether to apply it to our fields as fertilizer or dump it into bodies of water. In the developed world, we spent enormous resources dealing with this issue. Our sewerage systems and wastewater treatment plants constitute the largest investment of funds on any environmental pollutant that has ever been made. We have an infrastructure which is subterranean, largely invisible, and about which most of us never think. This success has been so broad that the very term waste, which at the beginning of the 20th century meant feces, urine, and other biological matter such as animal carcasses, has been repurposed to mean discarded manufactured materials. So the guts of the matter, in a sense, is an experiment. It investigates the changing spatial and temporal patterns of infectious intestinal disease. It's a very broad rubric that includes bacterial, viral, protozoal, and helminthic infections that are caused by dermal contact, in the case of some of the soil-transmitted helminths or worms, and the ingestion through a fecal-oral route of pathogens found in feces. In addition to historical textual sources, it draws upon a pretty broad range of scientific literatures, including the research of biologists, parasitologists, archaeologists, public health, and infectious disease specialists. There is no historiography on the global history of infectious intestinal disease, although there are some pathogens, such as cholera bacillus and hookworm, that have been researched at national or regional levels. The project, thus, is kind of a synthesis of different literatures in an effort to try and achieve new understandings of when, where, and how infectious intestinal diseases have been transmitted, and to explore the history of intestinal disease control efforts and the impacts of these control efforts on disease transmission. It's part of a larger project to create a scientifically literate global health history in order to create useful perspectives for those involved in contemporary disease control. Why is this important? Well, in a way, it's easy to make the case that infectious intestinal disease is important in the early 21st century. Let's try that one. Let's just take, for example, the case of intestinal worms called helminths. This is a map that shows the global distribution of soil-transmitted helminths. There are about 1 to 1.5 billion people today who are infected with one or more of the major intestinal worms: hookworm, whipworm, and roundworm. Viral and bacterial infections are major killers of children in the tropics. And indeed, the category of diarrheal diseases, a category that groups diseases caused by a large number of pathogens that cause the symptom of diarrhea, make up the single largest cause of childhood death in sub-Saharan Africa. And I guess you can see on this map how small the problem is in Europe and how it's part of the reason we don't think very much about it. The global burden of diarrheal disease is pernicious. The bacterial, viral, and protozoal pathogens typically rob the body of nutrients and fluids and cause severe dehydration and death. They exacerbate co-infections of children and often result in what we might think of as death by comorbidities. And some disease conditions are chronic rather than acute. In the past several years, we've had a new appreciation of a condition called viral enteropathy that's caused by repeated intestinal infections that results in an inability of the child to absorb nutrients and causes physical and mental stunting. Successes in controlling diarrheal diseases are one of the keys to understanding the rapid growth of some human populations in the last few centuries. And because rapid global population growth is one of the core drivers of environmental change, the historical epidemiology of infectious intestinal diseases can be understood as central to the field of environmental studies. Today my talk has three parts. The first is to suggest that the study of infectious intestinal diseases may hold some keys for understanding deep processes in human environmental history. The second is to sketch very briefly some new perspectives on intestinal disease generated by the Microbiome project in the biological sciences. And the third is to suggest some problems in our current understandings of global sanitation. So, part one, possible keys to deep processes. Our human ancestors in tropical Africa acquired a couple of the soil-transmitted worms, hookworm and whipworm, from our earlier pre-human ancestors. The fact which has been established pretty much by genetic studies, the fact that we were able to sustain chains of soil-transmitted helminthic infections suggests that human beings probably were not exclusively hunters and gatherers in the past. Hunters and gatherers have considerable difficulty in maintaining chains of infection from soil-transmitted helminths. It suggests that we must have had other life patterns, that we must have lived at least seasonally in coastal, riverine, or lakeside, lacustrine settlements exploiting fish and shellfish proteins. This is, of course, quite contrary to the accepted paradigm in history of human health, which holds that the first major epidemiological transition that took place was coincident with the agricultural revolution, when human beings achieved population densities that were large enough to allow for the transmission of certain pathogens. Other research on helminthic infections, worm infections, seems likely also to revise our understandings of the peopling of the Americas. The discovery in fossilized human feces, coprolites, of roundworm, one of the soil-transmitted worms that has a lifespan of about 1 to 2 years, They found evidence of roundworm infection in human coprolites dating back to about 8,800 years before the present. Those who work on the soils of the Americas are convinced that, I just took apart this light, excuse me a minute, are convinced that early migrants to the Americas, at least some of the early migrants to the Americas, could not have come overland, because it would not have been possible to maintain a chain of soil-transmitted helminthic infections. Some must have come by the hypothesized maritime route. Research on cultural practices to control infectious bacterial and viral diseases may also provide some keys to understanding deep patterns in human demographic history. If, as many historical demographers assert, intestinal diseases have been a principal brake on population growth around the world, it seems likely that the growth of Asian populations, even in the millennia before the Common Era, must have been facilitated by the cultural practice of boiling water. It seems unimaginable that the Shang capital in Anyang, established some 14 centuries before the Common Era and estimated to have a minimum population of 100,000 in an urban area in the second millennium before the Common Era, could have achieved this type of high population density without control over waterborne pathogens. So these are just three kind of elements of research in intestinal disease pathogen history that have some significance for rethinking early human history. Part 2, the Microbiome Project. Over the past several years, biomedical scientists have embarked on major research projects to investigate the human microbiome. Their research has produced new perspectives on the bacterial flora within our intestinal systems. One set of findings has illuminated the reality that different human communities do not have the same proportions of intestinal bacteria. In this respect, human beings possess rather different variants of a common microbiome. The different enterotypes are characterized by differently proportioned assemblages of bacterial genera. They may be controlled in part by genetic inheritance, although they're probably determined in larger measure by the kinds and quantities of foods that we eat. So in one sense, the research supports the facile truism that we are what we eat. There is a marked difference in the enterobacteria of those eating a diet high in protein and simple sugars, what in the microbiome literature is sometimes referred to as the Western diet, and those eating a diet high in complex carbohydrates. There are also marked differences between peoples living in intimate contact with the natural world. The overall bacterial diversity of the intestinal flora, for example, of the Yanomamo in Brazil, is much higher than the bacterial diversity of consumers of farm products. The composition of our intestinal flora reflects our diets, our varying exposures to wildlife and domesticated livestock, our hygienic practices, and our use of antibiotics. We typically think of genetic variation among human populations as differences in the allele frequencies of our Homo sapien genes. Yet because bacteria play such a significant role in regulating our basic metabolism of food and our immune responses to pathogens, We need to broaden our perspective to include the astounding genetic and metabolic diversity that can be found in the millions of genes and myriad of gene functions within our gut microbial communities. Our intestines are alive and engaged in vast and complex interactions among our intestinal bacteria and between our intestinal bacteria and bacteria elsewhere in our bodies and with our body organs. Strange as it sounds, microbiome scientists are coming to the view that we have two brains, one in our cranium and one in our intestine, and that there are complex communications between the two. We may think that the one in our cranium tells us what to eat. This may be mistaken. Our understandings of the interactions of bacteria, protozoa, viruses, and helminths are still rudimentary. The complexity of the problem is suggested by consideration of some round numbers. The bacteria in the human gut are estimated at an astounding 100 trillion cells in 500 different species of bacteria. The bacteria that each one of us is carrying around in our intestines weighs about 2 kilograms and makes up 30% of our feces. The intestinal bacteria in our guts produce vitamins. They modulate our immune responses to pathogenic challenges. They are critically important in our ability to fight off infectious pathogens. This last point has come into popular appreciation through the once unthinkable but now accepted procedure of using fecal implants to bolster the immune response to C. difficile, a particularly nasty bacterium that can cause devastating diarrhea in the elderly and others with compromised immune systems. Indeed, our ways of thinking about ecological associations between, for example, our gut bacteria and our body are becoming challenged by the new evidence. We used to think of, have a kind of a standard typology of ecological associations: symbiotic mutualism, commensalism, parasitism. Turns out to be pretty useful as a heuristic construct, but it greatly simplifies our understandings of our relationship to our gut flora. For example, the interactions complicate our understandings of parasitism. We know, for example, now that low levels of intestinal worms, such as roundworm and hookworm and whipworm, confer some benefits in enhancing immune responses to pathogens. We may be healthier with intestinal worms. It may be that the ridding of our bodies in the developed world of soil-transmitted helminthic infections has come at some cost to us. Yet at the same time, it's clear that higher levels of infestation by the same worms cause anemia, physical and mental stunting, even at the extremes, death. The degree of infestation makes all the difference. By way of biomedical metaphor, the dose makes the poison. Our intestinal universes have changed over time with our changing diets and with our species' expansion into new biomes. And our guts have had to adapt to our changing gut biota. Our guts reflect the physical incorporation of environmental influences to which our genes must necessarily respond. The study of environmental influences and genetic responses, that is, the study of epigenetic processes, will certainly open new paths for genomic study of the influence of the intestinal microbiome on nutrition and growth, and our understandings of pathogenic challenge and response. Part 3, global sanitation. Most of the infectious agents, viruses, bacteria, protozoa, worms, enter the intestines through the digestive tract. Some of the pathogens jump to us from the fecal matter of other animal hosts, including wild and domesticated animals. But most come directly from other human beings. The principal mode of transmission is fecal-oral, which means that typically we transmit pathogens with our hands by contaminating food and drink, or through touching our faces with our hands. Estimates of the frequency of this practice vary from 16 to 100 times an hour. In the 19th and 20th centuries, societies that experienced economic growth, the improvements in urban water supplies typically preceded the building of sewer systems that handled the water carriage of human waste. The development of piped water systems linked to sewer pipes created something new in world history: sewage. That is highly diluted human waste. Modern sewerage became one of the principal markers of modernity. There seems no doubt that waterborne sewage disposal was an aesthetic triumph. Our environments smell a lot better. But waterborne sewage has proved to be quite expensive, and from some perspectives, quite environmentally wasteful. In its early phases, it replaced a system of night soil, which is another term for human waste, night soil removal that transferred human waste from the city to the farm, and replaced this with an expanded system that dumped a large volume of sewage waste into rivers and lakes, contaminating the water supplies of downstream communities, and sometimes that of the discharging community, depending on water currents. The installation of the early piped water systems likewise produced unanticipated consequences. Once contaminated, the piped water systems had the potential to do something new, which was to spread pathogens widely through entire communities. We have lots of examples of this in northern India and in Egypt and elsewhere. The creation of public water supplies inadvertently made possible the poisoning of entire communities. Before this, you might have this type of infection contained within a family unit or within a village. With large integrated piped systems that became contaminated, it became possible to contaminate entire communities. This is a slide just showing you the types of vehicles that were available for purchase in the 19th century for moving human manure out of cities to the farms. This is a photograph from the late 19th century in Pennsylvania, just dumping night soil from Philadelphia onto local fields. Whoops. Here we have a photograph from the 1950s. This was the system of removing human waste in Warrington, which is a mid-sized town in the United Kingdom, about 60 or 70 years ago. And here is a photograph from 1911 in China. These are night soil boats that were removing the human waste from urban areas in China and taking them by canal to the agricultural fields. major industry in China and in Japan. The major successes in reducing infections from the contamination of water and food, the sanitation revolution in the industrialized world, has been in large measure the result of the chemical treatment of water with chlorine or ozone. In some areas, this has solved the problem of fully safeguarding the water supplies. It's been glossed as the achievement of improved sanitation and clean water. Today, clean water and improved sanitation are linked in a global public health discourse propelled by the belief that they're central to the control of infectious intestinal disease. The historical evidence, however, suggests that the volume of water available to communities is far more important than its cleanliness, and that an adequate water supply is far more important from a public health point of view than the creation of a system of sewage disposal. Some societies have dealt with human waste disposal, including open defecation in rural areas and the use of cesspits in urban areas and the transfer of urban waste to the countryside without major health problems. These two approaches, one of modernity and one of pre-modernity, if you will, remain in an unresolved tension today. So that's what I wanted to say. Those are my three topics. This is a very brief sketch of three topics that I'm grappling with in this research project. My larger goal is to try and contextualize the findings that have been made in a number of scientific literatures, and in the process to rethink some of the basic relationships between agriculture, sanitation, and health, as well as the deep periodization of epidemiological transitions. Thank you.